The coronavirus that causes COVID-19 has infected 33.5 million people worldwide, with over 670,000 cases and almost 17,000 deaths in South Africa. Admittedly, yes, South Africa has shown a highly positive recovery rate of 90%, and we feel we have beaten the virus. The global impact of this novel virus has nevertheless been severe, with some European countries now experiencing a second wave of infection. Patients that develop serious symptoms and recovered from virus infections are often left with long-lasting complications. Accurate testing remains important in controlling the virus and reducing its transmission. Currently, the PCR diagnostic test is the best method to test for COVID-19 infection, which is the test used at Next Biosciences. Dr. Holt, can you tell us about the COVID-19 testing being conducted at Next Biosciences?
Yes, sure, Charleen. We do diagnostic PCR testing by Thermo Fisher. PCR stands for polymerase chain reaction, and it tests for the presence of RNA of a virus. RNA is very similar to DNA, which we all know about. So a swab is taken from the back of the nose or the throat. In the lab, we swirl the swab in a bit of liquid, which releases all the cellular material collected on the swab. Then we extract the RNA from this liquid, test the RNA to see if the viral RNA is present. We test for 3 different RNA genes to know if the COVID virus is present in this certain patient.
Under South African patent laws, as in many other countries, a method of diagnosis practiced on the human or animal body cannot be patented. A diagnostic method and test is patentable where the diagnosis is actually performed on a sample and not directly on the body. In order to be patentable, the invention must be novel or new, and inventive or not obvious. While the PCR diagnostic test used by Next Biosciences is based on a well-known method, the test needed to include other new key features in order for valid patents to be granted. Dr. Holt, is the diagnostic test used at Next Biosciences a patented technology?
The test is a patented technology by Thermo Fisher. Their scientists developed this PCR test to test specifically for coronavirus. The test, as I said, tests for 3 specific genes on the coronavirus. Other companies around the world have developed their own patented tests too. Although all the tests are the PCR technology, each test varies in the balance of chemistry that is used, the genes that they test for, and the sensitivity and specificity. Therefore, each of these companies will market their own IP, their own methods to the scientific laboratories for the lab to choose which is the preferable test for them.
SAHPRA, the South African Health Products Regulatory Authority, has approved a rapid point-of-care serology test for COVID-19 antibodies and 3 lab-based serology tests. And if you are aware, SAHPRA has approved a rapid point-of-care serology test for COVID-19 antibodies and 3 lab-based serology tests. What is the difference between your PCR diagnostic test and these serology antibody tests?
Well, that's a very important question and something that everyone really should understand before opting to do either test. The PCR test is a diagnostic test. So that will tell you if you have the virus at that point in time. It detects the presence of the virus in the body. Your serology or antibody test basically tests if your body has produced antibodies to the virus. Now, how this works is when a human being is infected by a virus, our immune system produces antibodies to fight that virus. Initially, we produce IgM antibodies, so that's early on in the infection, 2 to 3 days after infection. And these are pretty non-specific antibodies. They are just there to identify the virus. Then the immune system makes IgG antibodies that are very specific antibodies, and they go out to try and eradicate the virus from the body. Some people, it has been shown, develop antibodies, others do not. There's still a lot that we don't know how our immune system reacts to an infection by coronavirus. So it is important to understand that just because you have not built up antibodies doesn't mean that you haven't had the virus. Antibodies may not last for very long. So they may only last in our bodies for about 3 months. Thereafter, the body stops producing them. The reasons for having an antibody test is really more for epidemiological studies, for us to see who's been infected, if they have antibodies, and who we should vaccinate. Otherwise, it doesn't necessarily give you a diagnostic result to say that you definitely have the infection.
Thank you, Dr. Holt. A COVID-19 patient may or may not present symptoms that require treatment. The symptoms have been of a wide range, with patients with particular comorbidities and elderly patients presenting the most severe symptoms. Scientists are racing to develop vaccines and drug treatments. In light of the urgency of the pandemic, the first approach has been to try drugs that have already been approved for other conditions. This is known as drug repurposing. Finding new uses for known drugs is vital to accelerating the development of treatments, as it speeds up the time it takes to make a treatment available. This is mainly because researchers don't have to repeat the earlier stages of development to show drug safety. A new use of a known drug can be patented if it is novel and inventive. We have heard a lot about remdesivir. This drug was originally developed to treat Ebola. It was shown to shorten the recovery time for patients hospitalized with COVID-19. Cipla and other pharmaceutical companies have obtained authorization from SAHPRA to import the generic of remdesivir under a Section 21 exemption. This allows for the import of medicines that have not been scrutinized by SAHPRA, but which have been approved by regulators in other countries. However, in August, the National Department of Health issued treatment guidelines where it decided to not recommend the use of remdesivir due to its high cost and marginal benefit. It has instead only been recommended for clinical trials. Oxford University is running a recovery trial to test whether existing drugs could be useful in treating COVID-19. It found that the drug dexamethasone reduced mortality among ventilated patients. Dexamethasone is a steroid that has been used since the 1960s to reduce inflammation in a range of conditions. The original drug is no longer under patent protection, but there are existing patents for related inventions. Dexamethasone has been in use in South African hospitals to treat critically ill COVID-19 patients since June, where it has reduced fatalities of patients in ICU by 25%. A key objective in fighting the pandemic is to develop an effective vaccine that protects people from contracting COVID-19 before they are exposed to the virus. There are more than 160 projects globally that are focused on developing such a vaccine. While the vaccine is only expected in mid-2021, this is exceptionally fast for vaccine development, which typically takes 10 to 15 years. Russia has already approved a COVID-19 vaccine, but there are concerns about its safety and efficacy. South Africa is hosting local legs of 3 international COVID-19 vaccine trials from Oxford University, Novavax, and Johnson & Johnson. Oxford's vaccine candidate has been shown to trigger a strong immune response. The trials have resumed after being put on hold due to a trial participant in the United Kingdom having an adverse reaction to the vaccine. The Novavax vaccine candidate is engineered from the genetic sequence of the virus that causes COVID-19. Studies show that it protects against COVID-19 infection in nasal passages and against lung disease. The trial in South Africa will evaluate efficacy, safety, and immunogenicity. The Johnson & Johnson vaccine candidate uses the common cold virus to deliver the COVID-19 protein into host cells, where it stimulates the body to raise immune responses against the virus. Research shows that a single dose of this vaccine protects against the virus. The South African trials will assess the safety and efficacy of the vaccine. Researchers succeeded in identifying the structures of the key proteins of the new coronavirus. This would normally have taken months or years to accomplish, but through open collaboration between researchers, it was completed in weeks. Open global cooperation is imperative at this time to arrive at a successful vaccine in an accelerated time frame. Now that international travel has been reopened and many restrictions have been lifted, we cannot become complacent. Until the wide global spread of the virus has been contained, we still need to rely on key measures such as social distancing, frequent hand washing, and monitoring flu-like symptoms. Take care and be safe.